Cara grew up in Massachusetts. She's from Situate, Mass, and went to Harvard for her undergrad. She spent a bunch of years doing field work and working for National Marine Fisheries. She was the sea turtle and marine mammal stranding coordinator for the northeastern U.S. for a number of years. And then I think in '07 started into this program. '05 started her PhD work at UNH, and since then her advisor Molly Lukovich, who's here tonight, has relocated to UMass. So Cara is now sort of in this dual UMass-UNH position, and started doing the bulk of the field work, I guess would be '08, '07 and '08. Most of the turtles we tagged that she's going to talk about tonight was an '08 field season, which was just an unbelievable year. So here's Cara Dubs.
Well, thank you all so much for coming out tonight. It's great to see so many faces in the audience. We've been satellite tagging, catching and satellite tagging leatherback sea turtles right here off Massachusetts since 2007, as Charlie alluded to earlier. And tonight I'm going to break up my presentation into essentially three parts. The first part will be some basic biology background, kind of letting you know how these animals tick, how they function. The second part will be taking everyone on sort of a virtual day in the field with our team. And the last part, I'll share some of the results of our satellite tracking efforts to date. So this is Leatherback 101, some basic biology. One of the most remarkable things about leatherback sea turtles that people always say is how enormous they are. They grow from these palm-sized hatchlings you see in this photo to adults that can weigh between 500 and over 1,000 pounds. The turtle in this picture is the largest turtle, leatherback, on record at about 2,000 pounds. And they reach sexual maturity in as few as 9 to 14 years. So these animals grow to these large sizes very rapidly. They also are quite unique among reptiles in that they're able to heat their bodies. Most reptiles are thermoconformers. Their body temperature matches the surrounding environment. But leatherbacks are able to actually heat themselves up. And they do this through a number of heat-conserving adaptations. They have a thick blubber layer that's very similar to marine mammals underneath their shell. And they also have countercurrent heat exchange in their shoulders and in their flippers. And countercurrent heat exchange, basically warm arterial blood that's leaving the core of the animal, heats up cool venous blood that's entering the core. And that way these animals are able to keep an elevated core body temperature of around 79 degrees Fahrenheit, which even up at latitudes such as ours, where water temperatures can dip below 60 degrees, they also travel to Newfoundland. They're able to keep their body temperatures over 20 degrees Fahrenheit elevated over their surrounding water, which is quite unique. They're also built for very deep diving. The pictures I'm showing here show a pelvis of a leatherback here and of a hardshell sea turtle here. You can see the leatherback's pelvis is in pieces, basically. They have what is called neotenic or unfused skulls and skeletons. Their skeletons are very loose and cartilaginous compared to hardshell sea turtles, which have a lot more ossified bone. And their shells are similar. This is a dead animal. There's no skin. And you can see these are the longitudinal ridges that run down the shell. And all these little dark dots you see are all these teeny tiny interlocking bones. Their shells are like a bone puzzle, all fitted together. And the reason it's hypothesized they have these features is because they dive very, very deep. These animals are capable of diving 3/4 of a mile deep in the ocean. So they need to be able to flex under this tremendous pressure. And this is they've evolved some very unusual mouths and gastrointestinal tracts. The picture here is showing a leatherback on its back. Actually, this is the upper jaw, and this is the lower jaw. And you can see there are two fang-like projections coming off the upper jaw, and then there's a fang-like projection down here. They have these very sharp scissor-like beaks. You can also see that their mouths are lined with spines. This is a view looking down into the esophagus of this animal. And they are covered, their esophagus is covered in downward pointing throat spines. In adult specimens, their esophagus can be over 6 feet long. And this is quite unusual, and you might wonder why any animal would have these features. And the answer is in their diet. These animals eat exclusively gelatinous zooplankton. So those sharp scissor-like beaks are used to grasp that soft-bodied, slippery prey and tear it up. And then the spines help macerate those jellies as they move down the esophagus. And when leatherbacks feed, they also ingest quite a lot of seawater. So they want to expel that seawater. They just want to keep the jellies. So those downward pointing throat spines hold the jellies in their esophagus while they're expelling seawater out of their nostrils. And these are actually two local flavors, I should point out. This is Atlantic sea nettle, and these are lion's mane jellyfish. And these are the, sort of the jellies that they seem to be queuing in on when they're feeding here off Massachusetts. And this is a video. It was taken in eastern Nantucket Sound a couple of summers ago. And basically we had just a simple camera attached to a pole that we lowered down into the water next to the vessel we were working from. There were several leatherbacks feeding in this area, and what you'll see when the camera lowers down is you'll start seeing a large abundance of lion's mane jellyfish. And what we were seeing is large numbers of these jellies, and they seem to be accumulating in these sort of drift lines of seaweed. Where different water masses are converging together. What you see at the surface is basically a drift line of flotsam seaweed, unfortunately a lot of plastic. These animals are also congregating in these areas because the jellies accumulate there as well. And they're able to very efficiently sort of pick off jellies under these lines of seaweed. And that's a lion's mane there. And what you'll also see in the video a little bit towards the end is we're also seeing lots of bells with no oral arms or tentacles on them floating around. We've never put cameras on our leatherbacks, so we don't know if they're actually adopting feeding behavior, but researchers in the Pacific have found that leatherbacks there are actually feeding exclusively on the oral arms and tentacles of jellyfish. This makes sense actually because that's the most nutritious part of the animal. The bells are mostly water and they're not very nutritious. But we don't know if leatherbacks here are doing that. We just see the evidence of these sort of empty bells floating at the surface. Okay, so that is a sea nettle. You can see they look quite a bit different from the lion's mane. So they're mixed in with the lion's mane, and you'll see shortly one of those jellies that's missing. There's the weed line that I was describing. Unfortunately, I don't see any turtles. They're kind of too far away. And there's one of the bells that I was talking about that's missing all of that sort of high nutrition oral arms and tentacles. See? So leatherbacks may in fact be doing that, adopting that type of behavior here. So that kind of gives you a little bit of basic background on how unusual these animals are. And now I'm going to take you into a virtual field tour, starting with introducing our field team. This is a small group of very dedicated individuals that have been with us since the inception of this project. We work primarily with commercial fishermen. This is Mark Leach, pictured here with the leatherback. And he is a lobsterman out of Harwich, Massachusetts. And George Permont, pictured here, is a spotter pilot. And these two have helped us find and catch leatherbacks for all years of our project. And we also partner with scientists. Charlie mentioned he's the veterinarian on all of our cruises. So scientists from New Aquarium, Charlie, Connie Marigo, who runs the rescue department here, has also been an integral part of this project. And we partner with scientists from Provincetown Center for Coastal Studies as well, particularly on animals that are entangled in fishing gear. So, most mornings, depending on where the turtles are located, we leave the dock very, very early. We usually get there to Harwich before first light, load up the boat. Usually winds are lightest off the Cape in the morning, so we try to get on site as early as possible. We need a very flat, calm sea state to do this work. And so we often get to see some rather spectacular sunrises as we're heading off of the dock. And as soon as there's enough light, we start setting up our gear. What we're doing in this photo is we're setting up the breakaway hoop net, which is what we use to capture the leatherbacks. You'll see that in the video coming up. And this needs to be adjusted throughout the day. We're always reattaching the net and trying to get the Velcro to stay on. It can be a little persnickety. And a lot of the equipment is set up as we travel out to our destination, which can be as close as 30 minutes away, or it can be as far as, sometimes we're going south of Nantucket, and it's a 3.5-hour steam out. As soon as we start getting close to our destination, we check in with George, our spotter pilot. There he is flying around his Super Cub. He leaves out of New Bedford and flies out to a predetermined location. We decide the day before based on where we've been seeing sea turtles, leatherback sea turtles, and where weather conditions will allow us to work. So once we're on scene, you can see these nice flat, calm conditions. These are both pretty perfect days for leatherback catching. We have to find the turtle, which is a lot harder than it sounds. It's definitely feels like looking for a needle in a haystack when you're out there. The ocean never seems so big when you're trying to find a sea turtle, even a 1,000-pound sea turtle. The leatherbacks in our area tend to come to the surface and breathe quickly and then dive right back down. We don't see a whole lot of basking behavior. So what we're looking for is usually just a head and a little bit of shell, and then it goes whoop, right back down. So everyone has to scan the horizon pretty much the whole time we're out at sea. We definitely get eye fatigue at the end of the day. And then we also have our person who's on the net. This is actually my husband does a lot of the catching, Mike Dodge. And he's at the net waiting in case a turtle pops up close to the boat because we don't want to miss any opportunity. And this is a sequence that shows 3 different capture events. It'll give you a good feel for how the hoop net works. So what we do is we approach these animals from behind. We've noticed, just based on the years we've been doing it, that they don't react to the sound of the boat, but they do react visually. So if they see the boat approaching them, they're very evasive and they dive and basically try to avoid us. So we have to kind of sneak up behind them and hope they don't see us. And then the hoop is lowered in front of the turtle, and there's a line coming off of it that then pulls the net off the hoop and purses the turtle up in a little net. Which makes it sound really simple, but it's very tricky. You can hear our spotter pilot directing us to the right, to the left, to the right. So some turtles are a little more cooperative and they'll be at the surface for a little longer. That first one was one of the cooperative ones, but most of the time we're doing this. We're sort of catching them as they come up for a breath, which makes it a little bit trickier. Definitely learn it. So of course I showed you ones that went well. There have been plenty that, plenty of turtles that we've missed, but those aren't quite as fun to show. It's definitely a learning curve involved with the hoop net capture, but we have a really good team that's gotten quite good at it now. So once we have a turtle in a bag, our next problem is how do you bring this behemoth turtle onto the deck of a fishing boat? We found that it works best when we work from commercial lobster boats that have an open stern. That way we're not having to get the turtle as far up out of the water to bring them onto the boat. And we've come up with a nice little system. We have a custom-built ramp that we lower from the stern of the vessel. And we've attached a salvage bag. Salvage bags are usually used for bringing up archaeological finds. We have one of these bags attached to the end of our board, and we inflate it with a dive tank, and that brings the ramp into a nice horizontal position. And then we are relatively easily able to slide the turtle and the board onto the boat. And some of them are really big, and it's not that easy. But it certainly takes a lot of the back-breaking work out by getting that the turtle up out of the water. And this picture I included, George took this picture from his plane, and it gives you a nice sort of perspective of the setup of our vessel. We've added a short bow pulpit to this lobster boat. That gives the net person some stability when they're out over. That net is extremely heavy, especially once it gets wet. It's quite awkward, so that gives the net person a little bit of stability. They have some railing system here that they can hold on to. And it gets us out over the turtle, so we don't have to get the boat quite as close to the animal. We want to avoid doing that. We also have the open transom here that you can see. Here's the dive tank set up with the hose. You can't really quite see the board in the salvage bag right there. And then we have the crew down here, and usually have at least 1 or 2 people situated on the wheelhouse to document, look for other turtles, that sort of thing. So, once we get one of these giant turtles on the boat, this boat is actually great because it has a transom that drops down, and then we're able to pull it right back up, because these turtles actually do wander around on deck quite a bit. And we usually just try to get out of the way because they're pretty large animals, and we just let them move around, and then once they're calmed down, we'll get to work. So this animal, as soon as we get them on deck, we remove that net right away. And we then soak them with, we have a continuously running saltwater deck hose, so we have to keep them nice and wet while they're out of the water. We also usually cover them with wet towels and sheets to protect their skin from the sun. We always cover their eyes with a wet cloth. This decreases visual stimuli and helps keep them calm. And everyone starts gloving up and getting ready to work. Charlie's back here starting the physical exam on this animal. We do a physical exam on every animal before we tag it. want to make sure they're healthy enough to be a tagging candidate. Okay, and this is another video. So this is Charlie. He's taking a pulse. And I'll be quiet so you guys can hear it. Maybe that whoosh. So that's the turtle's pulse. That's the turtle's heart rate. And he's doing this with a Doppler, handheld Doppler on the rear flipper of the animal. And this is super exciting work. This is only the second project in the world that's been able to get heart rates from these animals in their feeding grounds. So we're thrilled with how successful the New England Aquarium's health study has been, including getting these types of measurements. We also take a standard set of photos of every animal. Every leatherback has a pink spot on the top of the head. A lot of people who see them at sea think it's actually an injury, but it's actually a normal part of their makeup. They have individual, unique to each turtle, the shape of this spot. And so you can use this photo for individual identification, kind of like the fluke prints on a whale or a human fingerprint. So we always take a pink spot photo. We take 2 standard measurements of every animal, the curved shell length and width. Most of the turtles we tag, and you can see it's a 2-person operation to actually get these measurements just because the animals are so large. So here we're getting a width. We just use a flexible fiberglass measuring tape for this. And most of the animals that we've measured have been, the shell length has been about 5.5 to 6 feet. That gives you a good feel for how large they are. We also scan every animal for pit tags. These are passive integrated transponders, the same types of little microchips that people inject into their cats and dogs. These are routinely inserted into the shoulders of nesting females in the tropics. So we want to make sure, this isn't a known individual from a nesting beach, because the nesting beaches obviously would want that information. If there are no pit tags, we actually inject one of our own into the shoulder musculature. And this way, that animal's a female and she shows up on a nesting beach, they'll scan that turtle and be able to get back to us and say, oh, your turtle was here, which is really important information. And then finally, we get to our satellite tagging. Now, most leatherbacks in the past have been satellite tagged by attaching an external sort of backpack harness assembly. I'm not sure if people have seen photos of this. I don't have one myself. But it was quite a large, cumbersome piece of equipment on the animal. And Molly Luckcabbage, who's my PhD advisor, and is here tonight, she's been pioneering a direct attachment method to try to reduce this sort of hydrodynamic drag of the tag package to the animal. And so what we've been doing for our tags is we create 2 very small, you can barely see them, stick. There are 2 tiny little holes right there. They're about 4.5 millimeters, which is smaller than the size of a small drinking straw. And then we put surgical tubing through those holes, and then we thread biocompatible tethers with a corrodable link. And that then attaches the tag to the ridge bone of the turtle. And here you can see the tagging package complete on the animal. This pink, goopy, bubblegum looking stuff is actually a really cool product. It's a cold curing silicone putty that's actually used a lot for Hollywood special effects. But also works really well for this, because what happens is we're able to, each of the turtle ridges is usually a little bit bumpy or a little bit off, and this way we can actually put this putty on and conform that tag really well to the ridge, so it's not going to be flopping around, which is really important. We don't want that tag tearing off the animal. So this way we're able to get it really snug to that ridge. And you can see it's very low profile, just a little bit up off the turtle, and it's very small compared to the turtle's size. So that's really critical for reducing hydrodynamic drag. And that's not just important for the health and welfare of the animal. These are all endangered species. But it's also really important for, we're trying to collect natural behavioral data, and we don't want to have a lot of drag on that animal, because clearly then our data should be affected. So after we finish satellite tagging, the New England Aquarium team, that's Connie on the right and Kate Sampson on the left, they're back there collecting blood samples from the animal. Every turtle is blood sampled, and they also take culture swabs of the nostrils and the cloaca for their health study. And then we get to the sort of my favorite part of the whole procedure, which is releasing the animal back into the water. This is a very large adult male. Up here we tag males, females, and subadults. You can only access males in their feeding habitat. It's the only time the males are ever seen. They never come back to the nesting beach, so it's really unique to look at male leatherbacks. This is a particularly large one that was rather reluctant to get off the boat. So I picked this video because you get a nice view of the turtle making his very slow way off. It's funny, when they get on the boat, they're quite anxious to get off, but after about a half an hour of being on the boat, they seem pretty happy there. This guy was, he was really taking his time. So you see that's the drop-down transom there, so that's great too. The turtle's able to sort of slide right off of that. There he goes. Oh. There you go. So, of course, I'm like, I can't wait to get home and log into our account and start seeing where this turtle goes. I always love the release, and I get really excited to see immediately where they're going and what they're doing. And this last video, I just wanted to include one of the leatherback actually swimming underwater, because you see them on the boat, they seem really large and cumbersome, and but underwater they're just the most graceful, amazing animals. For such a huge, powerful animal, they just glide through the water, and it's just spectacular to see. Wish I had more of those videos. I don't. So now get to the results of our tagging work. So between 2007 and 2009, we put out 20 satellite tags on leatherbacks right here off the Cape. This map shows you the deployment locations. Most of the tags you can see, they were put out here in eastern Nantucket Sound. This seems to be a hotspot for leatherbacks in our area. We also put out quite a few tags on Nantucket Shoals, just south of Nantucket here, and a couple of tags in Vineyard Sound and Cape Cod Bay. Each of these tags collects location data on the animals, as well as information about where they're diving, the duration of their dives, and the amount of time they're spending at different depths and different temperatures. And the way these tags work is when the animal surfaces to breathe and the tag antenna breaks the surface of the water, the data is then transmitted to polar orbiting satellites above, which then transmit that data back to our data service provider in France. So all we have to do on our end is get onto a computer and log into our account, and then we have all of that data from the satellites. So it's really quite easy. All right. So I've displayed this on a Google Earth just because I think that's a really fun way to look at this data. This is the combined tracks of all 20 of our turtles. Each colored line is the reconstructed path of an individual leatherback. And as you can see, they traveled quite far and wide throughout the Northwest Atlantic. Our tags on average lasted for about 6 months. We did have a few tags that lasted longer and one tag that lasted for over a year. And the combined total dispersal of these 20 animals was over 90,000 miles they swam. So these guys are long distance travelers. We did not have any turtles cross over the Atlantic. We did have 2 that went over the mid-Atlantic ridge here, but then this one stopped transmitting, this one came back around. And no turtles went south of the equator. They all stayed right there in the Northwest Atlantic. We did see some differences between adults and subadults. So the adults tended to have more oriented headings. You can see these really nice sort of, even though they're widely dispersed, you see these very sort of consistent southwest headings as they're traveling. And mostly what the adults did, the subadults tend to have more variable headings, and they didn't travel quite as far south. You see here, this one actually went to the Bahamas. Which seems a little strange, but so the subadults maybe don't have quite as much experience. I don't know. So we did see those differences between our animals. And this map shows, the yellow lines show the exclusive economic zones of different countries in the area where our turtles travel. And as you can see, our turtles travel through quite a few of these exclusive economic zones, territorial waters. That's particularly here in the tropics, where they're going through, they're quite small. They're going through a lot of waters of several different countries. And this, of course, is important for conservation efforts because it just highlights the need to work internationally on conserving these animals because they're using the waters of so many different countries. And this bar plot actually will show you the amount of time they spent in these different areas. The y-axis here is the countries of the territorial waters, and the x-axis is the number of days spent in that area. And as you can see, by far, they spent most time in our study in U.S. waters and international waters, a combined total of about 2,500 days. But they did spend quite a bit of time in these other countries as well. Trinidad and Tobago looks to be particularly important, and I'll talk about that a bit later. Canada as well, Bermuda, Barbados, and Venezuela actually had quite a few turtle days. So our animals were getting out there, getting around. One of the objectives of our study was to identify seasonal high-use habitat. So we created these seasonal maps. There are 4 of them in a row, starting with summer. And what we did is we took the daily turtle locations and we summed them up into, it's kind of hard for you to see probably, but each of these little circles, they're actually hexagons. And we summed up the number of days turtles were in that area for that season. And the color bar runs from only 1 day in the dark blue all the way to over 60 days. And as you can see, based on this map, they had a very strong affinity to the northeast U.S. shelf in the summertime, and they were highly, highly aggregated. I mean, this is just a super hot spot right here, all the southern New England shelf and the shelf break. And then as we moved into autumn, turtles started expanding their distribution along the shelf. See here, there are still a few little red spots here in southern New England, but they start moving into the mid-Atlantic bight down here off North Carolina. And then they also started making their southward migrations in the fall, moving through subtropical and tropical open oceans. And their wintertime distribution was mainly confined to the tropics, and a lot of this has to do with breeding. These animals breed in the tropics. But as you can see, with very few red hexagons, you can see that they were much more dispersed, not as aggregated. There are a few little hot spots here, right off of breeding areas, breeding beaches in Trinidad and Tobago, the Tobago Basin. But otherwise, they were fairly widely distributed, and there were several turtles that actually overwintered here in open ocean, which I'll get to some interesting things they were doing out there in a bit. Now, unfortunately, most of our tags stopped transmitting in the winter, so we have very limited data for the spring. But what we did observe were 2 different behavioral patterns. We saw breeding animals remain in the spring here in the Caribbean, and then we had other animals that were not reproductive animals started moving north, and one animal that we tracked for over a year actually returned to the U.S. shelf and remained there for the entire summer. So another thing we were interested in doing was understanding how their behaviors might vary between distinct oceanographic regions. And by that I mean temperate shelf waters versus subtropical gyre versus the tropics. We're interested in how long they spent in these areas and what environmental features may have influenced their movements and their behaviors in these areas. So we tagged the turtles, of course, off of Massachusetts, and what we saw was they basically remained on the northeast U.S. shelf for quite a long time, an average of 66 days after we tagged them. And you can see that their movements are highly sinuous and clustered. And what this movement pattern tells us is that this appears to be a search behavior that's associated with foraging. So we're fairly confident these animals are foraging here, even though we're not directly collecting information about feeding. They also associated with cool temperatures when they're in this area, 61 to 68 degrees Fahrenheit, which again, remember, these animals are keeping core body temperatures of 79 degrees, so this is cool for them, 61 to 68. And they were associating with strong temperature fronts. If you recall the jellyfish video with the converged water masses in the seaweed, that's an example of some of these strong sea surface temperature front areas where prey accumulates. There was also very high chlorophyll A associated with the movements here, and I'll show you a map of that. So this map is showing the surface chlorophyll concentration for the month of August in 2008 when we were tracking 12 different animals in this area. Chlorophyll is a proxy for primary production, in other words, food availability. So high chlorophyll means there's probably more food there for them. And the color scale here is showing low chlorophyll in the purple and the blue, and the high chlorophyll is the warm colors, the yellows, the oranges, and the reds. And as you can see, coastal areas tend to be very high in chlorophyll, but also some of these areas here, like south of Nantucket, Nantucket Shoals, and here on George's Bank, are also areas of enhanced chlorophyll. And the reason for that is there's tidal mixing in these areas that mixes nutrients from the deep, cold, nutrient-filled waters up into those sunlit surface waters, and that allows phytoplankton to grow. So what we see when we overlay our turtle tracks on chlorophyll is that a lot of these very clustered movements tend to occur in these very productive high chlorophyll areas. And they also occur in areas where there's a strong chlorophyll gradient. In other words, where there's high chlorophyll meeting with waters that aren't quite as full of nutrients. As you might recall, we collected dive data as well from these tags, and what we saw in this area is that turtles, these sort of deep diving turtles, actually stayed really shallow. Most of their dives were less than 8 minutes long, and almost all of them were in the top 66 feet of the water column. So for leatherbacks, this is practically swimming at the surface. This is extremely shallow dive behavior. And what's interesting about that is that when you look at the average seasonal thermocline in this area, it's usually right around, average across the area is right around 10 to 20 meters or 33 to 66 feet. So it appears that these animals are actually staying primarily within the thermocline, which the thermocline being the warm surface water that overlays the cold water underneath. And what happens when water masses, that warm surface and the cold water underneath, come together is, again, prey will accumulate where those water masses are different. So they're probably staying warmer by doing this, as well as finding prey at that zone of accumulation. So moving into the subtropical gyre. So animals departed the continental shelf anywhere from the end of September through about mid-November, and they started moving through the subtropical gyre, also known as the Sargasso Sea. This area is known for being extremely nutrient deplete. It's not a very productive area. In fact, it's been referred to as an ocean desert. And animals behaved very much as if it was an ocean desert. They just wanted to get through it. We saw very directed, fast movements. You see very different from what you saw on the shelf. You see these straight line movements. And the animals were swimming twice as fast as they were on the shelf. And they stayed in this area for usually less than a month, 29 days average, associated with warmer waters here, over 72 degrees Fahrenheit. There were very weak fronts. There wasn't much frontal activity, and it was, again, very unproductive. Here's a map that shows you that. So you can see most of this area, other than here where the Gulf Stream is passing through, is basically the blue and the purple, very, very low chlorophyll. Not a lot of food for these guys here. When you overlay the tracks, you see that there are some somewhat sinuous movements here in that sort of productive Gulf Stream area, but after that, it's pretty much a straight shot. So dive behavior changed markedly when these animals left the shelf. They started making even shorter and even shallower dives, less than 4 minutes, and staying in the top 33 feet of the water column. But they also started spending over a third of their time at much deeper depth, so over 164 feet. And making longer dives as well, so over 28 minutes. And the reason for this may be related to the shoaling of that thermocline. During this time of year when they're passing through, the thermocline deepens, and they seem to be potentially targeting where that average seasonal thermocline is. We know that the surface waters are relatively unproductive here, so but it is probable that some of these deeper dives, they may be targeting some prey at depth where that thermocline is. But the length of time they spent there suggests this is not a very profitable habitat for them. And moving into the tropics, so all but one of our leatherbacks overwintered in the tropics. And you can see here there are some of these straight line movements moving into the tropical area, and then it starts getting more sinuous here when they get to sort of their destination. And the surface temperatures here, you might imagine, are quite warm, usually over 75 degrees Fahrenheit, often 80 degrees. And the chlorophyll is quite variable. I'll show you a map of that. Again, weak fronts. There are some stronger fronts on the South American coast. And they also appear to be honing in on some very large and dynamic ocean surface features, which I'll show some maps of. So here's the chlorophyll map for that area. You see kind of where they were moving quite quickly through. It looks relatively unproductive. But then there are these nice little pockets of productivity here along the South American coast and in the southern part of the Lesser Antilles. And indeed, when you overlay these turtle tracks, you see that these guys are no fools. Most of them are heading right into these very productive patches of water and not spending much time in that purple nutrient deplete area. And dive behavior, similar to the subtropical gyre, but they actually spent even more time at depth here. They spent over half of their time diving between 100 and 200 meters, or 328 to 656 feet. So they're making these very deep, long, over 32-minute dives in this area, as well as the short, shallow dives. Now this is an example of one of those surface circulation features I was referring to. This is a large warm core ring. This ring is moving in a clockwise direction towards the Caribbean. And the black line there is actually a turtle, a path of a turtle. And you'll see as I move through this, I'm going through one day at a time. The turtle follows this ring. It stays right in that ring, moves all the way around it. And these rings are basically associated with very enhanced productivity, particularly around the edges where the nutrients are being brought up. And this turtle probably very wisely chose to travel along that ring because it was able to forage very profitably there. And this also shows you, again, this is the most southernmost extent of several of our tracks in black. And you can see here, there's this area, this is actually not chlorophyll, this is sea surface height. It's showing that there's some very dynamic currents in this area. And what happens around 10 degrees north latitude is two different currents that are moving in different directions converge here, the north equatorial current and the north equatorial countercurrent. And when these currents converge, they actually mix nutrients from below up into the surface waters, and basically phytoplankton blooms occur. So this becomes a very profitable foraging area for not only sea turtles, but seabirds, marine mammals, and fish all target this area because of the enhanced productivity from this current activity. And I wanted to share some preliminary results from our genetics. We take a small skin sample from every leatherback we work on to try to figure out what breeding beach it's coming from. And we just got back some results. This pie chart shows the dark green and this sort of lighter green both refer to the Trinidad and French Guiana populations, nesting populations. And this isn't necessarily surprising because that is the largest nesting population in the Western Atlantic. So we know that they're putting a lot of turtles out into the ocean, and indeed you're seeing quite a lot of 75% of the turtles we've sampled seem to be from that nesting rookery. What's kind of surprised me a bit more was that we do have this nice little chunk of Costa Rica origin turtles. And one of our turtles that we tracked into the Caribbean Sea did end up nesting in Costa Rica and Panama. And just 2 years ago, we had an entangled leatherback that had flipper tags from Costa Rica. So it seems like we may have a fair few of these Costa Rica turtles in our waters. And we also seem to have a few of these Florida animals as well. And the reason this is important is because some of these populations are actually doing quite well. French Guiana and Trinidad, that population is increasing, as is Florida. But Costa Rica and the combined Costa Rica, Panama, Colombia population is actually declining. It's doing poorly. So it's very important to know where these animals are coming from. Unfortunately, we do have the dubious reputation of having one of the highest leatherback stranding rates on the U.S. East Coast here. Leatherbacks in our waters do run into quite a bit of trouble here. They get entangled in fishing gear, they get hit by boats, and they ingest plastic debris. So one concern we would have is if these Costa Rica turtles were we're having a high mortality of some of these animals that would be then seen as part of this decline on the nesting beach would be contributing to that. So that's something that we're going to keep taking more samples and hopefully finding out more. So, in summary, we saw leatherbacks aggregated on the northeast U.S. shelf during summer and early fall, and then we saw those migratory paths were very widely dispersed with those highly oriented headings as they went through the subtropical gyre. Their search behavior increases in cool, productive water masses where these surface fronts accumulate prey, and then that behavior seems to decrease when they get into these warmer, less productive oceanic habitats. And we're finding the ocean currents and some of these rings, like I showed you, the warm core ring, those types of features may be playing a very important role in the habitat choice of leatherbacks when they're down in the tropics. Dives in the northeast shelf were very short and shallow, and they seem to stay within the average seasonal thermocline. And the dive depth and duration increased as turtles left the shelf and moved into subtropical and tropical waters. We identified, at least for our population of turtles here feeding here, that they seem to have important breeding habitat in Trinidad, Costa Rica, Panama, and we saw some large adult males tend to sort of patrol the Tobago Basin, probably looking for females to breed with there. And international conservation efforts are absolutely critical for this species. So just a quick slide on future work. Unfortunately, this program is coming to an end. We would love to continue it, but we've been having funding woes, as I think most people working on endangered species do. We do have one more summer of field work coming up this summer. We are going to be putting our turtle tracks on seaturtle.org. I don't know if you've heard of that website. But we will be tagging hopefully 7 more leatherback sea turtle GPS tags and tracking those live online. We are also hoping to put out some very exciting new tags called daily diaries. These tags actually collect 3-dimensional behavior of leatherbacks in the water column, so we can reconstruct like exactly what they're doing for the first time instead of having this, just a dive profile. We'll actually have 3-dimensional behavior. So we're very excited about those. And this is my own little pet hope, is that we can someday get funding to do a prey characterization. We know, we're learning a lot about leatherbacks, but we know almost nothing about their prey, which of course we need to know a lot more to actually understand their behavior. And prey is sort of the missing link in all of this work. So I'm hopeful that someday we'll have money to do a study to actually characterize their prey using underwater video, some photos, and maybe even some acoustic technology. Many people to thank. We get most of our funding through the National Oceanic and Atmospheric Administration, and we also have some funding from the National Fish and Wildlife Foundation. And Cape Cod Commercial Hook Fishermen's Association has helped us out as well. This type of work, as you can imagine, takes a lot of people. I'm just the one lucky enough to be telling you all about it, but all of these people here, I won't read them all out, but everyone here has contributed in a very important way to this work. And of course, we have to get authorization through the National Marine Fisheries Service and U.S. Fish and Wildlife for working with these endangered turtles. And this is our website. Please visit our website, check out the work we're doing. There's more about leatherback research on there. I'm also doing feeding ecology work on the jellyfish, looking at what they're eating in stable isotope analysis. And other people in the lab are working on bluefin and big-eye tuna and sailfish. So a lot of exciting work to see on that website. Also link to all of our publications if you want to read more. And we also have a Facebook page if people are on Facebook and want to check us out on there. And that is it.